We previously discussed the importance of glycolysis. We noted that this is the central metabolic pathway that is responsible for converting glucose into pyruvate. And along the way, we invest a couple ATPs, but during the payoff phase, we yield more ATP. So overall, we generate 2 ATP molecules during glycolysis and 2 molecules of NADH, which is the reduced form of an important redox cofactor. So for each glucose that we use up, we produce 2 equivalents of pyruvate. In this video, we're going to think about what happens to that pyruvate when it gets metabolized to produce energy. Okay, so the first thing that needs to happen is that pyruvate needs to enter into the mitochondria. So before we really dive into that, let's think about the structure of the mitochondria. The mitochondria is made up of 2 membranes, the outer membrane and the inner membrane. The most remarkable feature of the outer membrane is that it contains a high concentration of proteins called porins, which facilitate the transfer of a lot of molecules across the outer membrane. Consequently, the outer membrane is quite permeable to a high degree of molecules. The inner membrane, however, is much more selective. Another really important feature of the inner membrane is that it contains a high amount of folds, which create these invaginations, or these kind of like these grooves when you look at it. And the reason for this is that it increases the surface area of this inner membrane. And that actually plays a really, really important role when we think about the chemistry that happens on this membrane. This membrane is the location of all of the electron transport proteins that are important in producing ATP. So by having these invaginations and having this maximized surface area, we can pack more of these proteins onto that membrane and have more chemistry that happens. Inside of the inner membrane is the matrix, and the space between the 2 membranes we call the intermembrane space, and we abbreviate that IMS. Okay, so now that we understand the anatomy of the mitochondria, let's now think about what goes on in the mitochondria. Okay, so as I mentioned, the pyruvate that gets produced from glycolysis has to enter into the mitochondria to participate in the TCA cycle. So the first thing that has to happen here is we take advantage of a protein that transports pyruvate from the cytosol into the matrix of the mitochondria. And once it gets inside, it's converted immediately to acetyl-CoA using this huge, really interesting enzyme called the pyruvate dehydrogenase complex. It's this conversion of pyruvate to acetyl-CoA which is the first example of an oxidative decarboxylation step. So in this process, the carbons of pyruvate get oxidized, we produce carbon dioxide, and in the process we generate one more reduced form of the cofactor NADH. So we produce carbon dioxide and NADH in this oxidative decarboxylation reaction. Okay, once we have acetyl-CoA, it can now enter into the TCA cycle. And we'll look at the chemistry that happens here in a little bit more detail in the next video. But the main idea is that the 2 carbons in acetyl-CoA combine with molecules in this cycle and then ultimately get decarboxylated to form more carbon dioxide. So the 2 carbons end up as carbon dioxide, and during this process we generate a lot of reduced redox cofactors, a lot of NADH and 1 equivalent of FADH2. Okay, so these are the really important electron carriers which will take the electrons that get produced during the oxidation of glucose. It takes those electrons to the next step, which is the mitochondrial electron transport chain. So these 2 reduced redox cofactors deliver electrons to the proteins of the mitochondrial electron transport chain, which move those electrons through protein centers, ultimately giving the electrons to oxygen to make And this overall redox reaction, where the electrons from NADH or FADH2 are given to oxygen, that's a really spontaneous process. So the energy produced during that redox reaction gets harnessed to pump protons across the inner membrane of the mitochondria. Okay, so protons from the matrix get pumped out into the intermembrane space. Now that reaction by itself, the proton pumping, is unfavorable. We are moving protons against a concentration and a charge gradient. Okay, so by building up that gradient of protons, we harness chemical energy that we can use in the next step to drive the synthesis of ATP. Okay, and the passage of electrons through these redox centers to build up the proton gradient, and then using this proton gradient to drive ATP synthesis collectively is known as oxidative phosphorylation. The absolutely critical enzyme that catalyzes the synthesis of ATP from ADP and inorganic phosphate is called ATP synthase. ATP synthase is this really amazing molecular motor that literally uses the energy of that proton gradient. It pumps the protons back across the membrane and quite literally moves a cog, right? It's like moving a wheel. It moves the axle of a wheel, and as you move this cog around, that drives the synthesis of ATP. So it's a really complex and really cool process that we will look at in a little bit more detail in the next several videos.